Okay, so welcome everyone. My name is Emilia Huerta Sanchez, and Sally Otto and I are co-organizing the software series, which is part of the hybrid thematic program on modeling and theory in population biology. And all of this is made possible by the Banff International Research Station. And so before I introduce our speaker, I want to remind you that we have another event next Tuesday at the same time. And it'll be an interview with Montgomery Slatkin. Okay, so let's start. So today I am delighted to introduce our speaker, Dr. Jerome Kelleher, who is well known for having developed msprime. Currently, he is a Robertson Fellow and a group leader in biomedical data science at University of Oxford. He and his group specialized in developing advanced algorithms and software designed to address challenging issues in large-scale genomics. Jerome's academic journey began at University College Cork in Ireland, where he received his PhD in computer science. And before assuming his current role, Dr. Kelleher was a senior statistical programmer in Gil McVeen's group at Oxford. And before that, he postdoced at University of Edinburgh's Institute of Evolutionary Biology and the Department of Statistics at University of Oxford. And so personally, I'm grateful to Jerome for figuring out how to make coalescent simulations computationally faster and for developing software that is powerful, practical, flexible, and makes, I think, our lives easier. And I just want to say that not only has msprimes become the simulation tool for generating genetic data on their various demographic histories, which can, of course, be used to benchmark new methods, to test hypotheses, and to develop intuition, but it also has inspired new methods development that leverage tree sequence data. And so in my lab, I know that we use msprime all the time, and I was just teaching before this, and we were using msprime there too, so it's also a great teaching tool. So with that, Jerome, thank you for being here, and the floor is yours.
Well, thank you. Yes, let me just get my slides shared. Okay, can everyone see my screen?
Yeah.
Fantastic. Well, thank you very much for such a lovely introduction. It's a real pleasure to be here, and thanks to the organizers for giving me this opportunity to talk about msprime. So, so the goals of the talk today, I'd like to give people a high-level sort of overview of the features of msprime and to highlight how to use these tools and how to provide some examples of their effective use. And with this in mind, I want to provide a few canonical examples of how you do sort of key things in msprime and how you integrate with the wider Python data science ecosystem using things like Jupyter, NumPy, Matplotlib, etc. And I also want to just point out a few common pitfalls for people who are just getting started with msprime or have been doing it for a while and may not be kind of structuring their code in the optimum way. So some non-goals. I'm not going to give an introduction into coalescent simulation because I think you can't really do this without giving an introduction to coalescent theory. And I think that's beyond the scope of this talk. And I'm not going to even attempt to be comprehensive about all of the things that msprime and tskit can do. I'm just going to sort of select a few things here and there that I think give a flavor of the possibilities. So an important question to sort of address before we even talk about doing coalescent simulations is, when should you use coalescent simulations and when should you use forward time simulations? So the downsides of coalescent simulations are that they are much less flexible than forward time simulations. So forward time simulations can accommodate essentially any evolutionary process that you can dream up. And there are some incredibly powerful and flexible simulators out there. And in particular, see the upcoming SLIM workshop from Ben Haller. SLIM is an amazing piece of software. The pros of coalescent simulations, though, is that they are very fast. So in the cases that you can model things, the things that you're interested in a coalescent framework, so you can think about things as being neutral or nearly neutral, then coalescent simulations are often several orders of magnitude faster than running forward time simulations. So I think a good rule of thumb for whether you should use a forward time simulator or coalescent simulations is you should probably use coalescent simulators whenever you can, because you'll save yourself a lot of time and some electricity. Another thing that I want to sort of address at the outset is that it's probably not obvious to people kind of coming in reasonably fresh to this, but msprime isn't a monolith. It's a component in a wider ecosystem of interacting and interoperating pieces of software that we've sort of been putting into place over the last almost a decade now. So these interacting bits of software include forward time simulators, so SLIM and forward pi 11 are very important parts of that. There's the software for thinking about demographic models, such as DEAMS and standard popsum that I'm going to talk about later. And then sort of the glue that's holding all of this together is the tree sequence toolkit, TS kit. So as a kind of a first approximation, the way to think about it is msprime is running your simulations and TS-kit is doing your analysis. So as soon as you get a result out of msprime, now you're in TS-kit doing your analysis. So jumping in, the main thing that we're interested in with a coalescent simulator is simulating ancestral processes, right? So the coalescent is a backwards in time model, and kind of by construction, it decouples mutational processes from the ancestral processes. And this means that, and through the way we do things in msprime as well, we completely separate the idea of simulating mutations from simulating the ancestral trees or the representation of the realized ancestral history that we get back. So there's a lot of kind of confusing terminology lying around. So we can call the simulated ancestry an ancestral recombination graph. We're not going to get into what we mean by an ancestral recombination graph here, but I'm basically going to use the term ARG, trees, tree sequence, more or less interchangeably throughout here. Essentially, it's the realized history of your sample. So we're going to do a bunch of these examples. So I want to show how you can do msprime simulations in a Jupyter notebook, and how you can use the available sort of visualization and introspection tools to effectively run your simulations and to understand what they're doing. So we're starting off with a very simple simulation here. We import msprime into our kernel, and we run msprime sim ancestry. We have a sample of size 2. I'll come back to what that means in a minute. And we specify an effective population size, NE, of 10,000. So we're simulating a very rough approximation of humans. And to make this example deterministic, I'm putting in a random seed. And then we get back our answer. This is the simulated ancestry that msprime has produced, TS. And then we use this really, really powerful and handy draw SVG operation on the tree sequence object to visualize it. So we say draw the tree sequence that we got back from msprime, and here it's a single tree. We can see that we've got four sample nodes at the bottom. We've 3 coalescence events. One happened at time 1500 generations ago. The other one happened at 2900, and then they reached their most recent common ancestor at node 6 at time 21,000 generations ago, whatever it is. So this result object is a tskit tree sequence, as I said. So this is a separate library from msprime. I'm not going to even attempt to talk about all the things that tskit can do, but essentially anytime we're doing analysis here, we're using the functionality in tskit. If you want to find out more about tskit, we've got a sort of website at tskit.dev, which tries to bring all of the different resources that are available together in a sort of easy to understand format. So there are different ways of looking at this tree sequence object. One is to just look at the actual thing itself in a notebook, and that gives you back this nice summary of what's in it. And the view has been slightly mangled here by the slides operation, but basically it's just telling us that we've got one tree, our time user generations, and telling us how many nodes and individuals and so on are in the trees. Another way of looking at it is this tabular representation. This is the underlying representation of the ancestry that actually produced, and that's moved around. So these are the nodes, and we can see that these nodes correspond to the samples that we specified at the present, and then we can see that the more ancient nodes here are these are the times that these coalescence events occurred. And then the actual topology of the tree is defined by this edges table. And that just says that there is a edge joining a parent, child 0 to parent 4 over a particular genomic interval. So it's very, very straightforward and simple representation of the ancestry. So a detail I brushed over a second ago is what we mean by a sample. So by default, msprime simulates diploids. And this means that if we take 2 sample individuals, we have got 4 sample nodes, because each individual is composed of 2 genomes, or ploids, or various things that people call them. And these are represented by nodes in the tskit model. And so we've got 4 nodes in total to represent 2 sample individuals. So to illustrate this, we can simulate other ploidies. So here we simulate a triploid. And here we say we still have 2 sample individuals, but now they each have ploidy 3. And so they correspond to 3 nodes, 3 sample nodes at the bottom of the tree. And to demonstrate how these nodes and individuals sort of relate to each other, I've set up some labels for the tree nodes. So we're labeling by I, individual ID, N, node ID. And so we can see that individual 0 here has node 0, node 1, and node 2. And then individual 1 is composed of nodes 4, 5, and 3. Under the hood, setting ploidy, it doesn't actually do very much. It just changes the time scale that the coalescence is happening at, but it's just trying to make these things easy to do, rather than sort of forcing the user to think about what the coalescent timescales are when you've got these tricky ideas of different ploidy levels and so on. Okay. So a key thing that we're doing when we're running coalescent simulations is we're sampling from a random distribution of trees or tree sequences or arcs or however you want to put it. And this random distribution is defined by parameters that we supply to sim ancestry. So in the examples before, I used this random seed argument to make the examples deterministic. But usually this is exactly what we don't want to do. We want to generate random samples from our distributions, so that we know that we're truly sampling from this distribution, and we're doing it in a reproducible way. So msprime works really quite hard to get you good randomness, and I think the basic message here is that you should trust it. So just to illustrate what I mean by drawing different random samples. We're running the same simulation again here without the random seed arguments, and we can see we get two completely different realizations, and that's all we mean by sampling from this. So the flip side of wanting to get lots of random samples is that we're going to need to do lots of replication in order to get at these distributions that we're interested in. So a single replicate from or a single draw from a random process with high variance doesn't tell you an awful lot. So we need to do lots of replicates, and msprime offers some specific machinery to make doing replicate simulations as efficient as we can. I'm just gonna demonstrate that there. So the sort of canonical example that should be can be used as a way to build up your own sort of simulation analysis pipelines is computing the mean and the variance of the time to the most recent common ancestor of a sample using 1,000 replicate simulations. And this is very straightforward. So we use NumPy here. So I definitely recommend using NumPy wherever you can. It's a very powerful numerical library. It's the sort of the foundation stone of the Python data science ecosystem. And we're going to do 1,000 replicates of our simulation, and we're going to use a NumPy array of length 1,000 to represent the the summaries of each of those replicate simulations. And the way we do replication then in sort of an efficient way is to pass this num replicates arguments to msprime, some ancestry, so keep the rest of the parameters the same, and pass this extra num replicates, and then we get back this reps value, and then we, in a for loop, iterate over these replicates. So this enumerate function here is basically just giving us a way to index the replicate ID. So it's not important that we understand everything that's happening here, but using this as a template for your own analyses is a good way to make sure that your simulation code is nice and efficient. And so for each replicate, we grab a tree. There's no recombination here, so we only have one tree. We get the time of the root. We put that into the jth slot of the NumPy array, and then when we're finished this loop, we output the mean and the variance of that array using NumPy. And this turns out to be a mean of 30,000 generations and a variance that's big. So I'm emphasizing this here because it's something that that people slip up on quite a bit, and their code ends up being quite a lot less efficient than it needs to be. I think it's a pretty common gotcha. So to illustrate this, we've done the kind of canonical example here in a timeout, and it takes 126 milliseconds, to run these 1,000 replicate simulations when we do it in the sort of recommended way. But if we do it in the in the same in distribution, so you get correct results by doing this, but it's 10 times slower. So the sort of obvious thing to do here if you're doing replication is to call sim ancestry 1,000 times in a loop, but it turns out that that's much slower than using this num replicates example, and it doesn't make much difference here. It's only a second, but this does scale. So if you've got a complicated demographic model and you've got lots of parsing going on inside Python, this can easily totally dominate the actual time to take the simulation, and it can be many times slower than it needs to be. The second kind of common gotcha here when people are doing this is that they store the replicate simulations in a list. And this is a sort of natural thing to think about, especially if you're coming from a language like R, where everything is a sort of explicit list. And you're not really used to thinking about these Python structures that represent implicit iterations. And so what happens here is, you get the same answer, and it's more or less takes more or less the same time, but it uses 1,000 times as much memory as it needs to, because it's storing all of these replicate simulations in memory at once, instead of looking at them one by one by one, as is done with the sort of the recommended approach. I've tried to illustrate this here with looking at these objects in the notebook. The first three elements of rep list are all tree sequence objects, whereas the original reps variable that we had a minute ago, that's a generator object, which is this Python tool for doing implicit iteration over a set. So takeaways here: msprime is good at randomness. You really don't bother generating random seeds and so on unless you genuinely need to. There's a lot of work behind the scenes to make sure that we're doing this well, and we're pretty sure it's doing it well now at this point. And for small simulations, using non-replicates and doing structuring analysis in a certain way can make a big difference to your CPU time and memory usage. So I'm primarily interested in talking about ancestry simulations here today, but I think it would be incomplete if we didn't talk about mutation simulations a little bit. So msprime is also actually a very powerful mutation simulation engine, and we can simulate mutations under many different well-known models. We added microsats recently, actually. And it can take any given tree sequence and impose a set of mutations on that. So it doesn't have to be something that's generated with msprime. It could be, something inferred with relate or TS-infer or anything. It doesn't matter. It'll impose mutations on that given the branch lengths and the parameters that you provide. So to illustrate this, we've run another ancestry simulation. Same as before, 2 diploids. This time we've said that the sequence length is 20 kb. So we're simulating 20 kb of non-recombining genome, and then we throw down mutations on that simulated ancestry at a rate of 10 to the minus 8 per generation, per site per generation. And then to visualize this, we throw this into the trusty draw SVG, and we can see then that we've got some mutations sort of drawn on top of our tree. And this is showing us the positions along the genome where the mutations occur. And then we've got the corresponding positions sort of on the branches where those mutations occur in the history. So it's a nice way of sort of quickly seeing where your mutations are happening, and so on. So if you, so there are lots of ways of analyzing sequence data in TSKit, but I just want to emphasize that you can get your data out in standard formats if that's what you need. So you can output to VCF, and then VCF can be basically consumed by whatever tool you want. I'll say that there's lots of functionality already in TSKit, so you may find that you can do what you need to in there, and that'll be much more efficient. But there's always the option of exporting to VCF and other formats if you need it. Okay, so population structure. We've assumed so far that we're simulating from a single pangmictic population that has had a constant population size for an infinite period of time. And this isn't particularly realistic. I don't think there are very many species that follow this. So usually what we're interested in doing is providing some more complex demographic models that that will sort of summarize, or at least reflect some details of the history of a particular species. So msprime has supports a pretty flexible model for demography. We've got essentially any number of discrete populations which have sizes that can change over time and migration rates between them. And it should efficiently manage pretty large numbers of demes. And rather than talking about msprime's sort of interface for defining populations, I'm actually going to focus here on a tool called demes, which is intended to be a standard for defining demographic models. So there's a paper in Genetics in 2022 if you're interested in finding out more details. But basically, demes is a declarative structure which defines a set of populations, their time existences and their relations to each other in terms of ancestry and also like migration rates between them. And the key idea is that if we have this single definition of what a demography is, then this definition can be used by the programs that are inferring these demographic models, and then we can transfer those inferred models directly into our simulators to simulate under them. And so demes is supported by quite a few different programs now at this point, and I think it's a really great. It's got some really nice tools for working with populations as well. So I'm not going to talk about how demes is structured, but basically we have three named populations here, A, B, and C, and we'll see what they look like in a minute when we use some of the nice tools that are available. So we want to define our demographic model. We do that here by importing demes and then sort of defining this YAML from the model that was in the previous page. So normally we'd just use a file. I'm putting it into a string here for convenience. And then we load up the that model into demes using demes.loads. So that parses this demographic model, make sure it all makes sense, and then returns you some object which represents that demographic model. And then we can do some really nice things with it. So one of the things we can do is use this lovely demes draw package, which lets us actually visualize the demographic model. So demes draw has this tubes function, which lets us do one particular type of visualization. So now we can immediately see that we've got two contemporary populations, A and B. B is smaller than A, and both of them merge into a single ancestral population, C, 1,000 generations ago. And this picture is worth 1,000 words of trying to describe what a population or a demographic model actually is. So coming back to msprime, how do we use this information? Well, msprime has its own demography API, and that supports demes as a sort of import format. And the way we do it is we've got this method msprime.demography.fromDemes that takes in the demes model that we loaded up a minute ago, and that turns that into an msprime demography class. And the demography object then has a bunch of features by itself, and one of the things it can do is provide these sort of introspection features when you're in a notebook. So when I'm in a notebook, I look at the demography. It tells me I've got these three populations called A, B, and C. It tells me their population sizes. It tells me about events that happened in the history of these populations. So our population split here is telling us that populations A and B were derived from C at that time. So now that we're working with a demography, there's another thing that we need to do, and that's specify where our samples came from, right? So now we're calling some ancestry with this demography object, which captures all of the complexity of a demographic model. And now we need to tell msprime where do we take our samples from, and the best way to do this is to use this form where we say we give the population name as the key in this dictionary, and the value then is the number of samples. So we're taking 2 samples from population A, 1 sample from population B. And again, we draw out this tree to see what it looks like. So this tree is handy. It it tells us some basic stuff about what has happened in the past of our simulation, but it's not giving a lot of insight into where events occurred. And we can use the information that's inside in the TSK tree sequence sort of model to allow us to get at that. So the TSK tree sequence does contain complete information about our populations, and it starts with this populations table. So these are the populations that were defined in the original Deems model, and they've come through as having names A, B, and C. And then the nodes themselves are associated with each of these populations, and there's various different ways you can get at this information, but I've just shown the nodes population array here. So this is saying that the first 4 nodes are from population 1, name equals A. Second 2 nodes are from population 2, which is name is B, and the rest then are from population 0, which is C. And then we can bring this information together so that we can improve our visualization. So now we can set up our visualization so that the node labels reflect the population in which the event actually occurred, and we can map that back to the metadata that was that was provided as part of the original Deems model. So now we can see that all of our coalescences happened in the ancestral population, which isn't surprising because it's such a small population. But still, we've got 4 samples coming from A, 2 samples from B, and then all of the ancestral nodes are occurring in population C. So the takeaways here are that there are some really, really good tools now for working with demographic models, way better than was available even a few years ago. So if you're still working with msprime 0.x demography models, then I'd really encourage you to make the switch over to the new demography APIs or even move over to using Deems as your sort of primary way of working with demographic history. I haven't talked about standard PopSim here, but basically if you're interested in simulating a particular species and that species has been added to the standard PopSim catalog, then that's the go straight to standard PopSim and use standard PopSim to simulate your species, because it's using msprime under the hood or SLIM, and it's trying to bring together all of these tedious details about estimated parameters from the literature into one place to make it easy to simulate particular species. So when should you use Deems and when should you use the msprime demography API? Well, I think basically Deems is really good for empirically estimated models. So when we're looking at these big complicated models that people are estimating for humans, for example, Deems is ideal for that. Whereas if you were working with simpler, more theoretical based models, then actually msprime's demography API is probably a bit more convenient. There's also a bunch of, I think, underutilized tools in msprime's demography API. So we've got, for example, numerical methods for calculating the coalescence rate trajectory for a given demographic model. And we can do really powerful things with this. I think there's really some really cool features in there that may not be used as much as they might be. Okay, so moving on from demography, up to this point, we've basically been simulating the ancestry of a single base of sequence, right? We're assuming that no recombination can occur, so we only ever get a single tree. And most of the time, that's not what we're interested in doing. So to make things a bit more realistic, we need to define 2 more parameters, sort of at a minimum. And these are the sequence length So that's the length of the sequence that we're interested in simulating, and the recombination rate, between base pairs along that sequence. So here we're simulating 5 kb at the human sort of rate of 10 to the minus 8. And when we do that, in this example, we get 2 trees. So recombination has occurred at position 601, and that's resulted in 2 distinct trees here, 1 tree to the left of 601 and 1 to the right. So I'm not going to go into the details of what all that means. Essentially, we've got these 2 trees, and now we need to do something with them. So visualization is very handy. It really helps us build intuition about what our simulations are doing, but of course it doesn't scale. So we can only really visualize these, sequences of trees with very short regions. So to illustrate here, I've expanded this simulation to just be a megabase, and even there we get 600 trees, which there's no way you can visualize. So we need to do something a bit more quantitative. So a very common pattern for this is to compute a statistic for each tree and to move along tree by tree. And TSKit has some very powerful ways of doing that. So the canonical example again of doing this is essentially to allocate a NumPy array, one for each one slot for each tree to contain the thing that you're measuring about that tree. And here we're looking at the MRCA along the genome for each tree. And we go through the trees in this loop, and then at the end we output the MRCA of that and the maximum of that for some reason. And so here that takes a millisecond to do for whatever 600 trees, which it's a small example, but it's still reasonably quick. And that tells us that our mean MRCA was 40,000 generations. So I'm not going to go into as much detail here, but there's another common gotcha here, which kind of results from people storing lists instead of using the iterators in the standard way. You get these confusing error messages, but basically don't take a list of the trees. You're just going to end up getting the wrong answer. So just trying to illustrate that again. And then one final example, just showing a nice way we can integrate with matplotlib in order to get a plot of something. This is the MRCA along the genome and the tree boundaries shown in gray, and we can do this with very little code, directly from the trees. So takeaways here, there's lots and lots of features in msprime for this sort of stuff. We have gene conversion, genetic maps, multiple chromosomes. The documentation is pretty complete on that. A thing to remember, and it's important to point out, is that msprime is fast for sort of low to intermediate levels of recombination. So human chromosomes are fine, we can simulate them, but for very high recombination rates, it's actually very slow. So trying to simulate a whole Drosophila chromosome currently isn't actually feasible. So if you're doing that, you're actually better off with a simulator like Scream or something like that, which uses the SMC. We're hoping to fix that soon, but at the moment, very high recombination rates do make msprime quite slow. Again, using TSKit effectively, following these sort of standard patterns can make a dramatic difference in how efficient your analyses are. And also there's a whole bunch of population genetic statistics in TSKit, in case you get the impression that you've got to build up these sort of statistics yourself by iterating over the trees. Most of the time you don't. Okay, so I've got one more big chunk of functionality I want to talk about. I'm going to go through that fairly quickly as I'm a small bit behind time. I think most exciting feature that's happened in msprime in the last couple of years is the ability to simulate trees conditioned on an input pedigree. And the idea there is that rather than simulating this kind of completely unconstrained process where we're using the coalescent model as a way of determining which lineages coalesce and recombine, we use this input pedigree as sort of tubes which the genetic ancestry gets shoved down and minced up and stuck back together. And the beauty of this is that the pedigree can capture the intricate details of history that are just impossible to model, you know, in a probabilistic way. So the models that are sort of appropriate on a continental level, it's very difficult to scale those down to the, you know, the population genetic processes that happen at a village level. And if we have observed pedigrees that are large and sort of detailed enough, then we can do just this. And that's the sort of thing that's demonstrated in this paper from last year, where we introduced this feature. It's some very nice results in that, I think. So quickly to show how this works, here's a pedigree. It's, you know, pedigree more or less like you find in various programs. We import this into msprime with this parse pedigree function. And then I'm showing the example pedigree just so we can visualize this. This function is defined in the msprime docs. It's using network X. What we get back is a tsket tree sequence that has the pedigree information encoded within it in its individuals table. So here we can see individual 0 has parents 4 and 5. We skip back here, we can see individual 0 has parents 4 and parents 5, right? So it's very straightforward encoding of the pedigree information within the overall structure. And then the node table also then shows how the different nodes for each individual relate back to these individuals. Same sort of standard stuff. Okay, so we then run this simulation with a slightly different pattern. We don't specify any samples, but instead we specify this initial state. So the initial state, this input tree sequence, which contains the pedigree information plus the nodes, you know, for the representing the genomes of the individuals in that pedigree, and the sort of the initial sampling conditions are taken out of that tree sequence by msprime and used as the way to set up the structure. The model then is the fixed pedigree. So when we do these two things, we've got an appropriate input pedigree and we say model equals fixed pedigree, then we simulate conditioned on that pedigree upwards through it. So again, we want to show what happens when we run this simulation, and there's with these labels, so we can see these individual 0 has nodes 0 and 1, and in this case, individual 0's genomes or nodes, they coalesce back in individual 9 at node 18. That's one thing to notice. The second thing to notice is that this ancestry stops at generation 4, right? So our pedigree only contains 4 generations of information, and what we do when we get to the top of that pedigree is we just stop. So these trees are incomplete. We have 2 different trees, and both of those trees have 2 roots, and the roots are the 2 nodes of individual 11, right? They're the same on both sides. The third thing to notice about this is that the history actually is incomplete. So looking again at the branch between 0 and 18, this has gone through the pedigree, right? It's after going through 3 generations of individuals and 3 generations of nodes. Now, no coalescence has happened, so we don't put in any nodes to represent that coalescence, but still, we have passed through known nodes, and actually leaving those out sometimes is a disadvantage. Sometimes we want to know the precise path with which the genetic material has traversed through the pedigree. But we can fix that now with these new features that have been added in version 1.3. These two new parameters called additional nodes and coalescing segments only. And with those two, we've got a very flexible way of specifying exactly what combinations of events are recorded by adding nodes on these long branches. So doing that here, we've got basically the same simulation, except we add these two new parameters, additional nodes and coalescing segments. And we're interested in basically everything here. So we're saying record all recombinant nodes, record all pass-through nodes, record all common ancestor nodes. And now when we do that, we can look at our branch between 0 and 18 again. And now we can see that we're after passing through nodes 11 and 12 by individuals 5 and 6 on our way through to pass through here. So we now have complete information about how genetic material has passed through the pedigree. And this, these arguments aren't only for the fixed pedigree model. You can do this with the Hudson Coalescent, DTWF, or any other model. And it gives you this information about where nodes have gone through branches where coalescences have not necessarily occurred. I think there's some really interesting work to be done in this area where we reason about what information is in these unary nodes and whether they can be detected from real data. The final thing to note about this is that the pedigree simulation just stopped at the founders. And usually we'd like to, simulate back to the most recent common ancestor of our samples. So msprime has got very powerful sort of mechanisms for doing this. And essentially we can add on an extra simulation on top of the simulation we've already done, telling it to complete it to ancestry. So this is the same method that we use for what we call recapitating forward time simulations. And there's a pair of papers here if you're interested in that, more details there. So here, our final tree sequence is, the output of our previous simulation. We're using the DTWF model here, which seemed more appropriate. And then we coalesce to a most recent common ancestor of everybody 6 generations ago. Okay, so the fixed pedigree model is very powerful. I think there's lots of exciting possibilities to explore there over the next few years, particularly when we combine with these large empirical pedigrees. And the ability to combine these multiple simulation types, even across different simulators, is a really, really powerful tool, which is well worth figuring out how to do. Right, so that's all I want to talk about. Thanks for listening. I just want to acknowledge my collaborators and the really big community of enthusiastic and fantastic developers that have sort of grown up around msprime and tskit. So these are the contributors from GitHub for msprime, and I want to thank all of them for their contributions. We have we're a friendly community, so if you want to join our Slack, or if you have any questions, please just do jump up on GitHub and post a question to the discussions board. We're always happy to help if we can. There's a bunch of publications if you want to get more details about any of these the ideas that are in here. So these are where the ideas were sort of originally set out. And there's quite a lot of online resources as well. I guess the main thing really is to use the tskit dev website, which has lots of links and information, and it's trying to put together these resources in a way that's handy to use. With that, I'm gonna thank you all for listening and stop sharing my screen. Thank you.
Thank you, Jerome. That was great. I guess we have some time for questions, if anybody has questions. Let's see.
What's the protocol for asking questions here?
All right. I think anybody can speak. I think David Petey has one, and then Ellen Backy. Sorry if I mispronounced your name.
Hey, Jerome. Thanks for the nice overview talk again. I guess I hadn't known that you could simulate multiple chromosomes. And going off of that, what is the best practices for trying to simulate like autosomes and like a sex chromosome concurrently?
Yeah, that's a great question. I don't think there is a best practice right now for the sex chromosomes. That's tricky. I think we haven't really figured out the right way to scale parameters and so on to do it properly. It's something we've talked about quite a bit in standard PopSim, but I'm not sure there was ever a proper solution. Yeah, doing the multiple chromosomes thing is a bit fiddly. You have to do a few things manually, which you can do it, and it's documented in the it's in the documentation.
Awesome, thank you.
Just
Ellen?
to say, you could do that in Slim if you wanted to, but of course it's not going to be as fast as doing it with the Coalescent.
Yeah, okay. Thanks, Jerome. I just like to ask whether there are any plans to extend to including selection, so things such as the ancestral selection graph.
So there is limited support for selective sweeps at the moment. So we have the basic machinery in there for doing the structured coalescence. But we're sort of, there's a few people are interested in extending that to make it more flexible so that we can kind of provide arbitrary allele frequency trajectories as the background of which you do the structured coalescent on. So I'm hoping that we will have sort of much better structured coalescent support in the, over the next year or so. For the ASG, I'm not sure. I'd have to scratch my head quite hard about that one, to be honest. That's tricky.
Yeah,
Sure, it's quite a different story because you have to add branching events and so.
Yeah.
Thank you.
Thank you.
Jasps?
Hi, hello. Yeah, thank you for the talk. I'm very interested since msprime is being used by so many people for so many different purposes. There must be a lot of thought, or it's like a very important decision what the default arguments are. So I wanted to ask you about what was the thought process behind making the discrete genome option being set to true, the default option for the simulations?
It was essentially through experience. So from the original defaults, the thought process was let's do what MS does because it's MS. And over the years of sort of working with people and seeing what they wanted to do and what they typically got confused by, that most of the time people actually seem to just want to simulate discrete genomes. Most of the time they want to be able to think about their study species, kind of simulate that as directly as they can without having to sort of, twist coordinate spaces and things around in their heads through, 4NE or whatever. So there was a conscious attempt with the 1.0 API to make things as simple as possible from that perspective. So, just do all of the coalescence, timescale manipulations and so on behind the scenes so that the users didn't have to.
I see. Thank you.
Thanks for your question.
Myra?
Hi, yes, thank you. And thank you for a very nice talk. My question is, so I had to simulate short tandem repeats before, so SDRs, surrounded by SNPs. And I know the way I did it was very, very slow. And I'm wondering if there is a better and faster way to do it. So basically what I did was, I gave the chromosome size that I wanted for the SNPs. And then I needed the STR like exactly in the middle. And then another set of SNPs. But when simulating the STRs, it created one STR per position. And so that meant I had to go through the whole generator to get the specific STR that I needed. So is there any way or a way of working on that would make it maybe faster?
I don't really know off the top of my head, Moira. I'd need to look at the details, but if you want to open a question on the GitHub discussions forum for msprime, be happy to sort of go back and forth a bit on that.
Okay, sounds good. Thank you.
Thank you.
I have a question. Or please speak up if you have a question, because I can't see everybody on my screen here.
Amelia, I was just pointing out that there was a request on the chat to if we could potentially get access to your slides, Jerome.
Well,
Sure.
okay.
I'm happy to give whoever the slides, but I don't know what mechanism that will happen by.
Okay.
So we will figure out how to post that maybe on the society website. Yeah. And I've just put a link in there. Or if you have it now, you can add it in the chat, Jerome, whichever.
That's sort of beyond my capability, Sally.
Okay,
You can email them to me, Jerome, and then I can post them on the web. I can send them to Noah to post on the website.
Okay.
Great.
I think there's
Thank you.
another question by Walid.
Yeah, hi. Can you hear me?
Yep.
Yeah, hi Jerome, how are you? Quick question. Very exciting the work that you're doing with integrating empirical pedigrees. I'm just interested in like picking your brain on what future directions you're kind of thinking about, because there's a lot of pedigreed wild populations out there. So yeah, I think it'd be very interesting to get those ecologists to kind of consider using msprime in that way. So what do you think?
Yeah, that's a great question. I think it gives us a way to, as I say, model this level of population structure that isn't really captured by the large scale, thousands of generations scale models that we usually think about. And it's a way of looking at the recent past and capturing the vagaries of the recent past, I guess, because when things are that detailed, the actual history matters, and the actual history is directly reflected in the pedigree. So if you go through this arbitrary bottleneck 3 generations ago, that's just there. It happens. So I'm not sure what the immediate application is. I guess that's not really my forte. But I think it's a new thing that people can do that wasn't really possible to do before, is to simulate, genetic ancestry and get out your full, the full tree sequence arc, whatever you want to call it, conditioned on this pedigree, and then examine, what are the effects of say, running multiple simulations through this particular pedigree. How constrained is the history by this pedigree, or how much variation is there around that? I think in the human context, looking at things like how important is this super fine-grained population structure on things like GWAS? There's a unique opportunity to do that now, I think. So I think there's some cool applications.
Well, it's 1 p.m. here. It's been an hour. Let's thank Jerome again for a wonderful talk. And I hope you join us next week for the next event. And so thank you for coming. And thank you, Jerome, for giving nice presentation.
Thanks a million for the invitation, and thanks to everybody for coming.